Okay, I'm just getting us rebooted to where we left off in Chapter 3, which was we were just wrapping up talking about membrane transport and the different types of solutions, active transport versus passive transport, and now we're going to get into a process that uses energy, the process of making proteins, called protein synthesis. The process of protein synthesis can be broken down into two main parts. You have transcription and you have translation. So first we need to understand the flow of genetic information in a cell. So I'll whip out one of my terrible drawings for you here. And the big idea is that DNA, which is found in the nucleus, is going to go through a process and create mRNA, which is a specific type of RNA, and that RNA is then used to make a protein. Remember, this whole process is called protein synthesis, so it's about making proteins. I need to get a better mouse. Sorry guys, this says protein. So the DNA, remember, is in the nucleus. That's transcribed into a substance called mRNA, or messenger RNA, and that messenger RNA is then used to make a protein. The process of transcription is what this step is all about. Transcription is DNA making mRNA. This occurs in the nucleus, which makes sense because that is where the DNA is located. DNA is like the big fat boss that can't leave the office, using mRNA as a messenger or as an intern, so to speak. The process of making mRNA into protein is called translation. And this happens at the little protein factories. Anyone remember what the protein factories are? The ribosomes. So this process happens at the ribosome. The first process happens within the nucleus itself. Now, one last thing, how do you remember the difference between transcription and translation? If you look at both of these words, they both have the word trans in them, or that prefix trans. Transcription has a C, translation has an L, C comes before L in the alphabet, so transcription comes before translation. So let's look at these in more detail. It says transcription is the instructions for creating protein being conveyed from DNA to mRNA. That's what I just reviewed. And then translation is when that mRNA carries the instructions to the ribosome. Another RNA molecule called tRNA is going to help with this process. It's going to bring in appropriate amino acids. You may recall that proteins are made of amino acids. So tRNA is going to match up specific amino acids from the mRNA instructions, ultimately creating a functional protein for the cell. So let's talk about transcription in a little more detail. The cell, of course, has to prepare for transcription. It's not automatically ready for it. So in the nucleus, the DNA has the instructions for making all of the proteins. Every single cell of your body has all of the instructions for making every single protein that you would possibly need. Nucleotides within the DNA sequence are going to specify the order of amino acids within that protein. If you recall, DNA is a nucleic acid made of nucleotides. Nucleotides are made of sugars, phosphates, and nitrogen bases. So those nucleotides come in 4 or 5 different varieties. And, sorry, that's my phone. Okay, so we were just finishing up talking about nucleotides. Let's go into the specific types of transcription. First of all, we have to understand one of the basic parts of this process. It's called a codon. A codon is a group of 3 nucleotides which are adjacent to one another within the mRNA molecule. This codon is going to code for a specific amino acid. So every group of 3 nucleotides within the mRNA molecule is going to code for a particular amino acid. The DNA can't leave the nucleus, as I said, so that mRNA is going to be made from a copy of the DNA. This is the process of transcription. To do this, RNA polymerase binds to a special part on the DNA molecule called a promoter. That RNA polymerase molecule moves along kind of like a train, just chugging along down the DNA strand, making a copy as the mRNA nucleotides pair with complementary DNA nucleotides. You'll recall that A binds with T on the DNA molecule and C binds with G on the DNA molecule. And so when there's an A on the DNA molecule, there would be a complementary T on the mRNA molecule. Except mRNA doesn't have a T, they have U instead. So if there's a C on the DNA molecule, there would be a G on the mRNA molecule. At the terminator region, that RNA polymerase is going to stop. It's going to dissociate and fall off of the DNA molecule and release the mRNA that has just been made. That mRNA now has a nucleotide sequence that's complementary to the DNA sequence. It has now been transcribed. It has the transcribed instructions for the amino acid sequence in order to make a specific protein. That mRNA is now going to leave the nucleus through those nuclear pores and go to the endoplasmic reticulum where it can meet up with a ribosome. Here's this process in more detail. You see we're talking about events happening inside the nucleus. We start at the beginning. Here's our DNA strand in chromatin form, this unwound kind of loose stringy DNA. You have the gene itself being identified. The gene holds all of the information for making the specific protein. The promoter is a region that's kind of upstream from the gene, and that's where that RNA polymerase is going to bind. That RNA polymerase chugs right down this DNA molecule, until it gets to the very end, which is the terminator sequence. In here, you can imagine that RNA polymerase adding these complementary bases, and you can see how they're complementary, A with T, and U on the mRNA strand with A, and G with C, and C with G, and so on and so forth, all the way down till you get to the terminator sequence, which marks the end of the gene. The process of translation is when the ribosome begins to attach to the mRNA at the start sequence. The start codon on the mRNA is the AUG. AUG signifies the very beginning of this specific gene. That ribosome is then going to move down the mRNA strand, just like the RNA polymerase moved down the DNA strand. In this case, though, tRNA, which is bound to a specific amino acid, goes into the ribosome, and those anticodons, which are a part of the DNA molecule, bind to the mRNA in order to correspond that amino acid that's on the tRNA molecule. So those anticodons on the tRNA pair up with the appropriate mRNA codons, and there's a special bond that forms between the amino acids called a peptide bond. The tRNA, which has disposed of its amino acid, now leaves, and then another tRNA comes in for the next codon, so on and so forth. Once the ribosome gets to the stop codon, which can either be UGA, UAG, or UAA, the ribosome is going to dissociate, the large and small subunits are going to break apart, and that newly synthesized protein is now ready to be finalized. Many ribosomes can be on one mRNA strand at the same time, so one mRNA strand can be used to make multiple copies of a specific protein. There's also post-translational modifications that have to take place. Once that mRNA strand dissociates from the ribosome, once the ribosome lets go of the amino acid sequence, that amino acid sequence isn't a fully functional protein yet. There's some other things that have to take place to finalize that protein. Here's the steps of translation. You have the beginning sequence starting right here, so you have some type of initiator. You can see A, U, G on the mRNA. mRNA is this little strand right here. This blue part is representing the tRNA molecule. There is an anticodon, which is the opposite codon on the tRNA molecule, and then each tRNA molecule also has an amino acid associated with it. So if our amino acid, or rather our mRNA, has the codon A, U, G as the start codon, our anticodon on the tRNA is UAC. It's the complementary to whatever's on the mRNA. Then there's a specific amino acid that matches up with that anticodon. In this case, it's the amino acid methionine. There are 20 different amino acids that could be used in this case. As the mRNA kind of moves along down, or rather through the ribosome, new tRNA molecules come in. Here's another tRNA molecule coming into the site. It's going to bind right here, the anticodon matching up with the codon within the ribosome. It's going to drop off an amino acid, and those amino acids eventually become bound together through those special peptide bonds, as I mentioned. And then over time, once the ribosome reaches that stop codon, in this case it's UAG, that will signal the end of protein synthesis, and the ribosome parts will break apart, and now you have an amino acid sequence that can be finalized into a functional protein. Here you can see what I mean, where several ribosomes are working on the same mRNA strand. So this guy's just getting started down here, but these guys are finishing up making the same protein. They're all just different steps of making the same protein. This video animation will show you some specifics on protein synthesis. I think it's a great idea to check out the videos because this is a pretty complex process. You can also try YouTube. Just be careful and make sure that you're using a .edu or something from a professor as opposed to a student's project that they submitted on YouTube. So we'll stop here for